and Balaji Chidambaram. Please come on this there, sir. Dr. J.R. Subramaniam is a senior consultant, internal medicine and diabetes, Apollo Hospital, Chennai. Has rich experience in dealing with various aspects in the field of internal medicine with specific reference to diabetology, and he has several national and international publications. Dr. Balaji is a director and diabetologist, Medway Hospital, Kumbakonam, and he is a member of Tamil Nadu chapter RSSDA, teaching faculty for various certificate courses in diabetes for doctors and paramedical staff, principal investigator for several trials in diabetes, successfully conducted Kumbhameton State Conference at Kumbakonam for the past 6 consecutive years, organized Track 2017 Tamil Nadu RSSDA Conference at Kumbakonam, and organized Kodavasi Manadu 19 Annual Conference conducted fully in Tamil, the first of its kind in the entire state of Tamil Nadu. Thank you. Please take over.
Thanks, Dr. Selva Pandian. Friends, Dr. Balaji and I have the onerous task of introducing this intriguing topic, the future of diabetes. Since the discovery of insulin, almost every 20 years, there has been a major breakthrough in the treatment of diabetes. Probably for the next generation, the insights into new islet cell technologies is probably going to give us the next level of treatment of diabetes. My colleague, Dr. Balaji Chidambaram, will introduce Dr. Anandkumar Annamalai. Dr. Anandkumar Annamalai has to catch a flight, so his questions and answers will be continued immediately after his lecture. Dr. Balaji.
Good afternoon to one and all. I am happy to introduce Dr. Anandkumar Annamalai, who is also a well-known speaker across Tamil Nadu and across India. He did his endocrine training at Anandbrooks Hospital, Cambridge, UK. He is a consultant endocrinologist, Ashwin Specialty Hospital and Vadamalayan Multi-Specialty Hospital, Madurai. And he has various publication journals, and he has written various things on endocrine book chapters. And he is the chairman of Educational Committee of Stem Cell Research Postgraduate Institute of Ophthalmology, Aravind Hospital, Madurai. Over to you, sir.
Thank you, sir. Good afternoon. Thank you very much for the kind introduction. So my topic is actually a futuristic topic. So it's a very onerous task actually to talk about something of the future and actually delve it into a dish for clinical practice. But my attempt is to try to simplify this futuristic topic, the new islet cell technologies, and labeling it as a curtain raiser for the clinician. This is the only slide which will have some form of different GIFs on this talk. So why are we concerned about type 1 diabetes specifically in India? All of us are concerned about type 2. Diabetes in children and adolescents, the Indian scenario is in fact also bad. We in fact rank close to the top 3 in terms of the number of children and adolescents with diabetes less than 20 years of age. So that's not a good thing to have. As of stages of diabetes, we now know that type 1 also goes through a lot of stages, and most of these basically are punctuated by the presence of the beta cell autoimmune response, which we can actually detect at all the 3 stages. Progressive beta cell loss occurs in relation to this, and we do get symptomatic type 1 diabetes at the later stages. Unfortunately, although we now have this pathogenesis, if you look at therapy in terms of therapeutics as stratified by the natural history, we do not have a lot. So this is actually the roadmap in terms of therapies directed specifically to this pathology. And what we find is it is actually very limited. In fact, there was a recent New England Journal of Medicine publication which was talking about monoclonal antibodies, which showed some promise. But in general, this is not very nice. So what are the conventional treatments? The conventional treatments are primarily medical in relation to insulin injections, multiple insulin injections, progressing on to an insulin pump, and then eventually then you go on to further definitive therapies, including a whole pancreas transplantation. Now, unfortunately, a whole pancreas transplantation basically has a lot of treatment failures, and 1 in 3 basically have big problems. So in the year 2000, further to the 60s when the pancreas transplant was involved, we had islet cell transplants with the Edmonton protocol invented in the year 2000. Now, this was an important landmark in terms of therapy. So if you look at the roadmap, this is how we started. So as I mentioned, multiple daily doses, continuous subcutaneous insulin injections, and then beta cell replacement in forms of transplantation from the pancreas or islet cells. And then as we moved on, and we will talk about, includes stem cell islets, which are derived from various stem cells, xenogenic in terms of other animal cells, and also other aspects which include beta cell regenerative techniques. So, as I mentioned, the roadmap towards islet cell transplant, the landmark was in 2000. The pancreatic transplant per se has been ongoing since 1960, whereas the islet cell transplant took off since the 2000, since the Edmonton Protocol. So although we don't have a lot of this, in fact, practically none of this in India, in certain specialized centers in the West, islet cell transplantation is done. The indications primarily are people with type 1 diabetes who are receiving intensive insulin therapy, and in spite of this, basically they have a poor glycemic control with an A1C of less than 9, with a presence of a low C-peptide, which has to be very low, with basically treated diabetes-related complications and no malignancies, because obviously, this would be counterproductive in terms of a transplant. You could categorize people into whether they have preserved kidney function or a kidney function which basically has been altered and you have had a transplant of the kidney, or if you have completely altered kidney function. So if you have a preserved kidney function and the age is between 25 to 35, and you have a labile hyperglycemia, meaning the glycemia basically is up and down, and you have plus or minus the presence of hyperglycemia unawareness with at least one severe hyperglycemia event during the year, then islet cell transplant alone is indicated in this cohort. If you have someone who has already a kidney transplant with a stable function, a stable renal function, with which I mean an eGFR of greater than 30, and an albuminuria which is less than 0.3, you could consider an islet after a kidney transplant. Or if someone's had a pancreas kidney transplant and the pancreas has failed, then you could consider an islet cell transplant. So this is in relation to someone who's had a previous kidney transplant with a reasonable eGFR. What about people with type 1 with end-stage renal disease? If you have end-stage renal disease and the eGFR is less than 30, the preferred option even now is a pancreas kidney transplant. However, if this is not desirable, you could consider doing a simultaneous islet and kidney transplant. So what happens here? So you either have an allo transplant or an auto transplant. An allo transplant is a pancreas from a donor, and an auto transplant is the very own pancreas retransplanted. The allo transplant, although the islet cell transplant you know about, is still considered experimental, although a lot of countries have got approval for this. The important issue to understand is you're actually transplanting islets, and you require at least 600 to 1,000 islets per kilogram body weight, which in fact equates to 2 to 3 diseased organ donors. The auto transplant is not for type 1 diabetes. You could use it for people with chronic pancreatitis, especially when you're performing a total pancreatectomy or a distal pancreatectomy. The steps for islet cell isolation includes a host of things right from the pancreas harvest, the organ preparation, the isolation of the cells, the purification and culture. Now the purification and culture are important steps. Now why is this so? Because the islet cells have immune potential, they're immunogenic. And if you actually purify it, then the immune response comes down. However, by doing so, there is loss of viability. Because if you say you take n number of cells and you're going to culture it, you're going to lose some of the cells in a kind of plan to actually make it more purified. So these cells are then taken and then they are introduced as an infusion. So you're not actually transplanting an organ, you're actually infusing cells. Now how do you do it? You do it with two sources. Transmesenteric, using a general anesthetic, or transhepatic, using percutaneous approach, which could be done under local anesthesia. So this islet is infused, and whilst the infusion is happening, you're actually also going to immunosuppress because you don't want immune rejection. And where are you actually injecting it? You're actually injecting it into the main portal circulation because that is the site where you would want the insulin to be acting and not in the systemic circulation. And to prevent clots, you're actually also going to give heparin. So what about these islets who actually are used after culture? So as I mentioned, after culture, the immune suppression actually comes down. So that is actually advantageous. Procedure complications obviously include hepatic bleeding and portal vein thrombosis. The entire process involves all these steps as we talked about. And the immunosuppression was actually, classified very well by the Edmonton team. And the Edmonton protocol actually, started this particular combination of treatment, which includes a T-cell activating inhibitor, an mTOR inhibitor, and a calcineurin inhibitor, which actually made those cells actually function well. Do islet cell transplants have good outcomes? This is the question which everyone has. And if you look at trials, across whatever has been done, this is the summary of the trials which have been done across the globe. Now, if you look at this is the 5-year data for islet cell transplantation alone, that's the ITA, or islet associated with kidney transplant, or simultaneous islet kidney transplant. What you can see is the 5-year insulin independence is close to 40 to 50%. What about A1C of less than 7 and no hypoglycemias? This is close to 60%. Do we have any data in India? That's one of the important aspects. We have one particular trial which was done from the Asian Hepatology Institute in Hyderabad, where basically there was a distal pancreatectomy with an autologous islet transplantation in subjects with chronic pancreatitis. Remember, this was not an allotransplant, this was an autologous islet transplant. However, if you look at centers which are actually evolving into this, Postgraduate Institute of Chandigarh, and there are certain studies going on in Apollo Hospital, Chennai. So these are the centers which are actually looking into this actively. So the islet cell transplant successful outcome. If you look at all the favorable outcomes, as I mentioned, the 5-year transplant survival, as the transplant survival, not the survival of the individual, was close to 70%. And the 5-year insulin independence was 47%. And the most important aspect as far as microvascular and macrovascular outcomes were much better in the islet cell transplant arm. So with all that, we still don't have all this yet here. So why do we need a new islet cell technology? So the islet cell transplant, although it is new for us, it is not new to the world because it has been happening since 2000. So it's 20 years, we are in 2020, and that is not a new islet cell technology. So why do we need this? And that is because the pancreas and islet cell transplant require a large number of donors, and it has certain procedural complications we spoke about. The immunosuppression still has to occur, although it is just a procedure. It's not a big transplant procedure. It is, immunosuppression is still needed. And when you immunosuppress, you get infections, predominantly also involving, viral markers, which can predispose to certain cancers, specifically into the skin. Kidney damage can occur with the immunosuppression. And both autoimmune and alloimmune responses can occur, which obviously is important. So what new islet cell technologies are then needed? There are 3 important aspects to look into. One is to develop a renewable beta cell source. And number 2 is to find ways to maintain these beta cells, because you don't want these beta cells to damage. With the islet cell transplant, over a period of years, it weans down. So it's like any transplant, you would need further injections. And the most important aspect is to shield these cells from the immune attack. Because if we have a mechanism where we are able to kind of shield these cells, then we don't have to give these immunosuppressive agents. So renewable beta cell source, we know the allogenic islets which we are doing. The xenogenic islets predominantly are obtained from pork, from pigs, the porcine source. Now with the porcine source, the problem again is, do we get enough pigs? But now with genetic engineering, that is not a problem, as with insulin. Now we have genetic engineering techniques where we basically have a lot of insulin-producing islet cells from the porcine source. However, there is still one problem, that you can still have hyperacute rejection in certain situations. But again, this is being modified in a fantastic way across the world. There is the theoretical risk of zoonosis with retroviral infections, especially with animals. Going with what's happening across the world in China right now, the porcine, xenotransplants basically, can create a problem. So what are the other renewable beta cell sources? The human stem cells basically can be used to produce insulin cells in an unlimited supply. You could use embryonic stem cells, however, that has certain connotations with regulatory agencies, mesenchymal stem cells, pancreatic stem cells, hematopoietic stem cells, and fetal stem cells. So what do you do with these? So these are the potential sources for new beta cells. And these new beta cells can actually, in the lab, in vitro, evolve and develop close to a beta cell which basically produces insulin. Now the problem here is those beta cells have to be harvested and in fact have to be transplanted again into the particular person. So there again, you have certain problems in terms of immunoreactivity. So the other aspect is if you have pluripotent stem cells, and if you have those cells derived from mesenchymal cells, you're not going to have rejection. But what about off-target differentiation of these stem cells? You are destining them to go into insulin-producing cells, but what if they become pancreatic ducts or pancreatic acinar cells? So there is a potential neoplastic hazard with this. So here comes the novel technologies which basically are coming into being. So now we have something called neo-islets, which is basically a durable stem cell-based biological therapy, which is combining a mesenchymal stem cell and a beta cell in a way that basically the beta cell is in the center and the mesenchymal cells are around it, and this actually prevents the immune basically action and prevents the immune rejection. The other aspect over the period of years are gene replacement therapies. So where gene replacement therapies basically modify these particular areas and actually make these immune tolerant. What about the ways to maintain a beta cell and actually prevent an immune attack? One is the site. So a lot of people question, why actually transplant the islets in the liver? The simple solution is we want a portal circulation of insulin, and that's the reason why we do it. But that obviously can create problems. What about anywhere else? So if you're going to inject it somewhere else, let's say in the muscle, or if you're going to do it like a subcutaneous tissue, like insulin injections, the problem is these islet cells have to survive. Now, the islet cells are dependent on oxygen, and they're also dependent on vascularity, and they have to be permeable towards insulin secretion. And remember, the insulin secretion has to be best delivered into the portal circulation. So how about going into this? So this is a novel technique where they're using tissue engineering of pancreas. What they're doing here is basically they're retaining the structure of the pancreas, they're digesting the enzymes, they're keeping the scaffold of the pancreas, so the basic structure remains, they're digesting everything, they're in fact zapping everything out, and then they're repopulating this with stem cells in terms of islet cells. So this is, you basically have a core, you deplete the entire content of this, and then you refill it with genetically engineered stem cell derived islet cells. So this basically would not have a problem, because you could actually put this into your omentum and you could actually have a similar portal circulation. So this is where we're heading towards in terms of novelty. The other ways to prevent an immune attack is in terms of immunomodulation, by using novel therapies as to how you basically make the immune system work better in concordance with preventing immune attacks. The other aspect is immunoisolation. So you're actually isolating these islet cells which you're actually transplanting, and you basically shield it from the immune cells which are being produced through technology. And how can they be achieved? By a process called encapsulation. Encapsulation could involve two processes, microencapsulation and macroencapsulation. How does this work? So this is basically a kind of schema of how things work. You have the islet cells in the center. You have a semi-permeable membrane around it, which can be a device, it can be a coat, it can be anything of that sort. It basically is the islet cells are able to secrete insulin and get the nutrition from outside, but basically is shielded from the immune system coming from outside, and that's how it works. As far as how you do it, it could be a medical device. So it's like, you have a kind of a core. You could microencapsulate, you could peg, you could cause conformational coating. And these nano and microencapsulations basically protect it. Or you could have devices. So you could have devices which are basically wrapped, and it's actually basically, the cells are put in, and you could insert it in multiple places inside the body. And this basically has been proved in a lot of animal studies and actually some human studies to be very effective in terms of producing insulin, which is responsive to your glucose. So this is the xenograph based on porcine islets using microencapsulation, and this basically was able to shield off the immune attack. The macroencapsulation basically devices a large number of islets, so meaning that you don't have to use, you have to use only a limited number of these inside the system. You could use 2 or 3, which is called Therasite or Encaptra, or you have this beta O2 device. All these devices basically have ways to prevent the immune attack and promote the oxygen content inside these particular compartments. So the PEC NCAP is one of a very novel candidates, which was developed by an agency called ViaCyte, and they actually use human pancreatic progenitor cells created in the lab. They basically, implant these into patients where these mature into functional pancreatic islet cells, and the encapsulated cap basically prevents and protects from the immune system. So this is the ViaCyte device, which I spoke about, which basically is a recharging encapsulating system, which is in the size of like a mini card. And this can actually be put into the system. Another great breakthrough which came out recently was from Harvard. And this is a pancreas on a chip. So you basically have, Harvard scientists have found this, where you basically have a chip system, which basically is an islet biosensor. And this is not just going to be limited just towards type 1 diabetes. This is for even drug therapies for type 2. So if you're going to look at the efficacy of a particular drug in terms of how it's going to be acting on the pancreatic islets to be secreting insulin in response to glucose, this is the chip. The chip on the right-hand side actually has two chambers. On one, the cells are basically lined up. You get insulin and glucose basically pushed into it, and the response is measured, and it gives you a readout. And this is very, very amenable to the clinician. So this is going into for FDA approval very soon, and this is a novel application in the islet cell technology. So a lot of trials looking at the beta encapsulation system in clinical trials are in phase 1 and phase 2. As I mentioned, this is a futuristic talk, but this basically is the way we are heading towards in terms of delivering these novel technologies in terms of treatment. So if I were to summarize, and I want to look at all what we talked about, so the beta cell replacement therapies involve all of these. The allotransplantation, which we spoke about, the beta cell regeneration, whether it is due to stem cells, the xenogenic islets from porcine, the immunomodulation and the immunoisolation from basically the various immune shielding techniques we spoke about. So to conclude, islet cell transplant has long-term efficacy in the last 20 years in preventing lability of diabetes and complications. Access to islet transplantation is limited due to unavailability of donors and immunosuppression. And novel islet cell technologies look into alternative sources of islets, extrahepatic sites, and immunoisolation. Islet cell technologies and the future. It's a long way to go, but as from what this Chinese proverb says, the journey of 1,000 miles begins with a single step, and the first few steps make a big difference. Thank you very much.
Friends, this topic is open for questions now. We can take 2 questions, please.
So the attempt actually in terms of looking at rodent studies and animal studies has been done in the South, by the Asian Institute of Gastroenterology and Hepatology. So they are the pioneers in terms of looking into this. As I mentioned, the 1-year successful trial of an autologous islet cell transplant. So it was not an allo transplant, it was an autologous islet transplant. They used it in people who had chronic pancreatitis-related diabetes. And they found the 1-year data being very successful. They had 4 patients, 3 of which actually had a good improved C-peptide at the end of 1 year. So that was a trial which I put up. The other centers which are actually actively involved in this now are Postgraduate Institute of Chandigarh, which are actually, they're looking into this very actively. And also Apollo Hospitals Chennai has got some program in relation to this. But in terms of DCGI approval, in terms of a trial, it is Asian Institute of Gastroenterology which has done it.
If there are no other questions, may I request we move on to the next topic.